Our target is to develop a very stable microbial consortium comprising of bacteria, funguses, and all other things, and then that consortium can be sustained in a very a way that in which it can be handled easily into, as you mentioned, oil spills. Oil spill has been historically a very grave problem which is prevalent across the world. Our country has also witnessed the Mumbai oil spill, Chennai oil spill. So the oil spill leads to very significant damage to the environment. So when it comes to the coast, when it comes to the soil, our target is to prepare a material that contains the microbial consortium immobilized onto it, and then that material can be used onto the soil or the sand or the polluted area, where that consortium can assist into easier or faster degradation of those compounds. So that societal concern is always there. We are working towards developing a consortium that is very robust and can not only degrade simpler hydrocarbons but polycyclic aromatic hydrocarbons. So that is our motto.
And for example, soil, water, it's everywhere. And I would like to believe that most of the areas it's polluted. So how many of these devices or this consortiums would be needed to solve this problem at a larger scale?
Yes, actually, the consortium development does involve a huge collection of diverse type of samples. Like if we are a petroleum laboratory, we will emphasize only on the petroleum spills, either the terrestrial spill or the marine spill or any kind of other spills which is accidental. So when you say that everywhere it is polluted, so we would say that where there is a requirement, where there is urgency, there it can be used at priority. Otherwise, these microbes can be, microbes can be used in different forms to all the areas where it is polluted. But our concern is to provide this material to a high priority polluted area.
And we do not have anything of such kind at the moment.
We have some products. But still the validity needs to be ensured for wider application. So we are just improving if anything is available. Of course, it's available globally, but we are on to improving their efficiency and make a robust this consortium.
My last question to you would be that since you're using fungus and using these natural things that exist in the environment, like natural beings that exist in the environment, do you believe that that's a way we can actually solve pollution or tackle climate action in a way?
Dr. Sunil, please.
Yeah, actually, in environment there are microbes and fungus are already present, and they are doing their work for remediation. But as I mentioned, that their pace of working is like for their survival only. They are not, working for us. They are working for themselves. So there are microbes, but the thing is that we have to identify such microbes and the more potent microbes that can degrade the pollutant in faster way. So that's why in the microbiologists are searching lot of microbes from different geographical area. The meaning is that the geographical area is very important, because the microbes which is working here that may not work on another site, because their growing condition is different, because their survival condition is different. So it is very important for microbiologists to select a vast range of microbes and make a consortia for treating these pollutants. So this is in this direction we are also working. Apart from this polyaromatic hydrocarbons, we are targeting the pollutants which are coming from fertilizers, the pollutants which are coming from the apart from the petrochemical industry. We are also working on that, but that is on initial stage. Our students are working on that.
I'd also like Sunil to mention about the secretomic part. because enzymatic, and we have already identified some enzymes. So, if you'd like to mention the secretomic part.
Yes, because the microbes for degrading any pollutant or any compound, xenobiotic compound, they use their machinery. And what is the machinery? Machinery is their enzymes. They produce a lot of enzymes inside that, and they take the pollutants and then they break into the smaller compounds through various metabolic pathway. So we prepare the consortia and check in presence of the xenobiotic compound what that microbes are producing in that environment. Because suppose we are trying to degrade X compound, and the microbes doesn't have the potential to degrade X, so there is no use of that microbes in that consortia. So before doing any experimental plan for further broad study or in real time study, we are studying the secretomic pattern of that microbes, how that microbes are behaving in presence of certain pH or any pollutant. Is that system available with that or not? So that is also we are taking care of that secretomic study. So how the pollutants are affecting their metabolic pathway, or the gene, how the gene is regulating, it is upregulating or downregulating, and what kind of secretory protein they are releasing in the environment. So this kind of a study we are performing right now.
And what would be like the kind of collaborations or things needed for you to actually take what you've developed to, for example, actually like into the oil spills or the marine spills that we call about?
First of all, we need access to the polluted matrix. The real world pollution, as you said, into the marine spills is accessible. And to develop a consortium beforehand, so we need to have an access to the sites where the spills is actually taking place. For example, if a pipeline is going from a refinery to a certain distribution terminal, so there is a leakage and there is a terrestrial spill, we need an access to that microbial consortium or the microbes present in that environment. Then when a crude is imported, it goes into the refinery. When during its handling it is spilled into the soil, we need access to those matrices. So when it is distributed at a retail outlet, it is again spilled on an ongoing basis. So accessibility of those areas is the first thing. If that accessibility is easy, I must say it is right now not easy to go and find a polluted site, and that too with an access to handle it, to see it and to collect it. So that is easier, than that formation part of the scientific study planning is more easier.
Adding to this, on collaboration side, apart from this thing, I mean, knowledge is also required from the different areas. Suppose this is not just microbiology, and it requires a lot of bioinformatics things. It requires a lot of analysis, chemical analysis. Like that, Dr. Pankaj is chemical analyst, and myself is biotechnologist. But the interdisciplinary knowledge is very much required, because the application of biotechnology in environment is difficult. I mean, not easier than other fields. Other fields have already, developed a lot of things. In agriculture, biotechnology application is there. In pharmaceutical, a lot of biotechnology application is there. But in environmental field, it is still growing. So we required a lot of knowledge from the different area. This collaboration of the knowledge is also very much important.